so much, Wendy. And now we're going to go proceed with our next speaker, with Rick Blumberg.
Yeah, thank you very much to the organizers. This is a very important meeting, and I'm really glad that I could participate. I think it was Wendy who invited me and asked me to talk about atopic disease and the microbiota, and that's what I'm going to talk about. So I'm going to start with this first slide to bring clinical relevance to what I'm going to tell you about, which is something that has been called the atopic march. The atopic march is the notion that there are a variety of allergic diseases that affect especially children. And they they occur with a different age distribution. But the reason that this is called a march is very often these diseases are seen one after another in the same child, suggesting that there is a common pathway, if you wish, that is leading to the susceptibility and the development of these diseases. And a hypothesis, this has been noted for many, many, many years, and a hypothesis that has been put forth and has been very popular as a hypothesis to explain these atopic diseases and the so-called atopic march was first coined by David Strachan, published in the British Journal, a medical journal in 1989, when he described a cohort of children and their development of these atopic diseases. And he observed in this very important paper the observation that there was an association between the atopic march, if you wish, and the household size in which the child was born, the birth order, so that children that were born first were more likely to have the atopic disease as opposed to those who were born later. I'm a firstborn, so I'm really in bad shape. Explains a lot of things besides the atopic march, as well as hay fever. And it's very interesting. He described this in a birth cohort were all born during a single week in 1958. Subsequently, Jean-Francois Bach wrote a very important commentary in the New England Journal, which hypothesized that complex diseases that many of us study, including myself, inflammatory bowel disease, multiple sclerosis, and so forth and so on, rheumatoid arthritis, are increasing throughout the world. And at the same time, they're occurring in association with a decreased microbial burden. Again, consistent with the notion, and he utilized a hypothesis laid down by Strachan to explain this increase in complex diseases, which altogether suggested that exposures early in life were affecting the later life susceptibility to these atopic diseases and these complex diseases. And some epidemiologic evidence has subsequently been put forward by a number of different investigators. I'm just choosing one study from Erika von Mudius in Munich, which was a New England Journal paper, which showed that there's an inverse correlation between asthma and growing up on a farm, which correlate with exposure to a wider range of microbes to the children. Again, consistent with the notion that early life exposures, the proper early life education by the microbiota, was providing some kind of protection or lack of protection thereof in children during early life. A disease that we study, many of us in this room study, and looking at many of my colleagues around the room, is a disease called inflammatory bowel disease, although not really an atopic disease, although there are shared immunologic characteristics and pathways that are involved in IBD as there are in atopic diseases. We don't consider IBD to be an atopic disease, but rather a complex disease that's polygenic and associated with very important environmental influences. But also, IBD, like many of these complex diseases, and also like the atopic diseases, are increasing in incidence in early life, especially during the first decade of life, and have been linked to, at least epidemiologically, to early life exposures to factors such as antibiotics, which affect the microbial exposure, and have therefore led to the notion that early life exposures or lack thereof may affect later life susceptibility to inflammatory bowel disease. So some years ago, we began to think about this. One thing that we do in my laboratory, been doing for a very long time, is studying a very interesting group of T cells called natural killer T cells, which I feel like we're going to have a little bit of an immunology talk here now, and if you want me to shut up, I'm happy to do that, but I'll continue on. I see some smiles, so I'll continue. But we've studied NKT cells, natural killer T cells, and we, for a number of reasons, we considered the possibility that NKT cells may be under the influence of early life influences. But in order to do this, and this is my animal welfare slide, the only way we could actually address whether early life exposures as opposed to later life exposures, which we felt were critical for providing biologic plausibility for the hygiene hypothesis as seen through the lens of NKT cells, would require germ-free animals. And I just refer you to a wonderful review written by Andrew McPherson, who's going to be giving a talk here tomorrow morning. And I'm looking around, I don't see Andrew in the room. But I'm looking forward to, as all of us are looking forward to talk tomorrow, that the only way to answer this question was to utilize germ-free animals, where we would expose them to microbes in early life and look for a biologic event, and show that exposure in later life that was not able to confer that phenotype. That would be biologic evidence that early life exposure would have an impact on the host that would be consistent with what might be considered necessary for biologic plausibility for the hygiene hypothesis, with the notion that if early life exposure provided a particular immunologic event that was protective, then the animal would be resistant to a later life trigger of that particular biologic pathway, that is that later life trigger of natural killer T cells, as opposed to receiving the education event from the microbes in later life, which would not have that property. So let me give you a little bit of a primer on NKT cells so you understand the pathways. NKT cells are a third group, if you wish, of T cells that instead of recognizing peptides in the context of MHC class 1 or class 2 in the case of CD8 cells and CD4 cells, NKT cells recognize lipid antigens in the context of a non-polymorphic class 1-like molecule called CD1d. There are two flavors of NKT cells. The major one that we study, although not necessarily the major one in terms of the numbers of cells in humans and mice, but the one that is more tractable is the so-called invariant NKT cell, which carries a semi-invariant T cell receptor alpha chain and can easily be studied with CD1d tetramers. These cells are very important in early immune response as orchestrators of downstream events. We have been studying NKT cells for a long time, and one of the several of the things that we discovered over time that led us to this notion that NKT cells may be important here is we realized that NKT cells are very important in regulating bacterial commensalism, whether it's with a pathogen, as you see on the left, or with a commensal, as you see with E. coli, Lactobacillus gasseri, and some other organisms that monocolonized CD1d deficient germ-free mice had a very difficult time managing that commensalization. In order to study NKT cell biology, in addition to looking and examining their ability to manage commensals, there is a very useful model that was actually established by Warren Strober's group some years ago, who's a senior mucosal immunologist at the NIH. Warren discovered that a hapten, oxazolone, when given to mice, will induce and trigger NKT cell activation in a CD1d restricted manner, that if you remove NKT cells or remove CD1d, these mice do not respond to oxazolone, which has become a very, very useful model for, as an environmental, if you wish, trigger of NKT cell activity, and a very useful model for assessing what I'm going to show you phenotypically shortly in terms of the ability or inability of microbes at conventionalizing at different phases of life to regulate NKT cell numbers and activity. And NKT cells have also been linked to inflammatory bowel disease in humans because NKT cells can be observed in the gut of humans with IBD to have increased production of interleukin-13 as an example. And NKT cells, when you isolate them and do gene arrays, are enriched in genes associated with genetic susceptibility for the disease. So this is really the interesting experiment that very talented postdoc Torsten Olzak did, which is the basis for everything that I'm going to show you. And I'm going to use my laser pointer, and I checked my compass and when I was sitting on the other side, and this is the north. And since nobody on that side of the room has received any attention for this last morning, I'm going to move to the north, and this is the south, just to let you know. That, as you can see here, all the folks in the north, that if you colonize, if you look at the numbers of NKT cells in the colons of individuals, of mice that are colonized under conventional conditions, they have a certain number of NKT cells in the colon, and the germ-free mice have an expansion of the NKT cells in the colon. And you can only normalize that expansion of NKT cells in the colon if you colonize or conventionalize those mice pre-weaning. If you conventionalize those mice post-weaning, you cannot normalize the NKT cells. This has turned out to be quite interesting and quite reproducible by a number of laboratories around the world. And what's even more interesting to me as someone who studies inflammatory bowel disease, I'm going to go back to the south now because I can see it easier. That if you expose a germ-free animal to oxazolone, which is a trigger of NKT cells, as I told you, that you can see here that the animals that are conventionalized in neonatal life gain protection from exposure to oxazolone in later life, whereas the animals that are conventionalized in a later life and have this massive expansion of NKT cells in their colon are not able to be protected by this later life environmental trigger. And all the data on the right are just bells and whistles of controls, and they're really not important. The important point is, in order for an animal to not respond in adult life to an environmental trigger of NKT cells as modeled through oxazolone, utilizing the observations of Warren Strober, you have to give the microbial signal during the first couple of weeks of life. Now, another model is asthma. It's part of the atopic march, as I said. Interestingly, asthma is another disease that has been linked originally by Dale Ametsu's lab to the biology of NKT cells. We find the same thing, or we found the same thing in the lung, that when we looked in the lungs of animals that were germ-free, that there was an increase of NKT cells, and the numbers of NKT cells in the lung could only be normalized if they received the conventionalization during the first couple weeks of life. Not only was that the case, but the animals then as well, when exposed to a model of allergy using ovalbumin hypersensitivity, exhibited, and it's well known that that model is related to the activity of NKT cells. That's been published by others. That it was very, very obvious that, again, only the animals that received the colonization and the commensalization during those first couple weeks of life, but not thereafter in later life post-weaning, were able to receive resistance to the development of asthma. So two different diseases, one an atopic disease and one a complex disease, could only be normalized in terms of their sensitization to later life triggers of those diseases if they received the microbial education during the early part of life. And when we looked at the mechanism, we found that at least one of the mechanisms was due to microbially induced repression of a chemokine ligand, CXCL16, that was very important in NKT cell recruitment. And as you can see here, I'm going to try to move to the north again. If you look in the CXCL16 levels in the animals that were conventionalized, they have low levels of CXCL16. If one looked in the serum of animals that were germ-free, they had elevated levels of CXCL16. If you looked in the animals, they can't hear? Okay, sorry. I'm going to have to stay south, guys. I'm sorry. Apologize about that. If you look in the animals that were conventionalized in adult life, they maintained those very high levels of CXCL16, and only again if they were conventionalized during this window of opportunity, as we've called, are you able to normalize the CXCL16 levels. You see the same thing if you look at CXCL16 in the lungs and the colons, and we were able to observe this was due to microbially induced epigenetic repression of the CXCL16 gene. And if you inhibit CXCL16 with antibodies in early life, you can abrogate this entire pathway. And a real-life example is actually antibiotics, which has epidemiologically been linked to a lot of these diseases, that early life antibiotics would theoretically remove some magically important microbes in the host that are important for this early life education. And so in this particular experiment, if you take SPF mice and pregnant SPF mice and expose them to antibiotics so that the pups are born into a germ-reduced environment and maintain the antibiotic pressure for 2 weeks, you can see that the mice become very susceptible to the oxazole-induced colitis in a CD1d dependent and NKT dependent manner. So antibiotic reduction in early life can also reveal this phenotype. It's not just germ-free animals. A very important set of observations, and I have to say that we couldn't have done any of these studies if it was not for a very close collaboration with Dennis Kasper's group, who maintains a germ-free facility in his laboratory at Harvard Medical School. Dennis has been studying Bacteroides fragilis for many years, and Bacteroides are one of the few bacteria that are able to make sphingolipids. And a very talented postdoc in Dennis's lab, Ding Yang, took this observation and generated a strain of Bacteroides fragilis that was unable to make sphingolipids by deleting an enzyme that's the critical first step called serine palmitoyltransferase. And very interestingly, that when Ding did the same colonization experiments, she could find again that this is again looking at the SPF animals. This is the level of NKT cells in the SPF colon. There's this expansion in the colon in germ-free animals. If you monocolonize with a wild-type B frag, you can normalize this. And if you monocolonize with a B frag that's singularly unable to make this sphingolipid, this is disabled. And Ding went on to show that, again, you had to give the B frag in neonatal life in order to, you had to give the B frag in early life in order to normalize this, and similarly to convert the animal to resistance to the oxazole in later life, one had to give, again, the B frag in early life. All this is consistent with the notion that early life colonization, even with a single organism, is able to induce resistance to later life triggers of NKT cells through normalization of NKT cell numbers in the colon. To go one step further, again, the talented chemist in Dennis's lab, Seung-Won Oh, went on to actually isolate what one might be one of the critical factors, one of the critical sphingolipids that are important in this pathway. Seung-Won Oh was able to isolate a galactosylceramide from the B frag that had the abilities to suppress and inhibit the ability of NKT cells to proliferate as one of the potential mechanisms that was conferring this resistance. So what I've told you today is that early life exposure through the lens of NKT cells is very critical in normalizing NKT cell levels in the animals, and therefore regulating the sensitivity to later life triggers of NKT cells. And this again is to show that if colonization occurs after the so-called neonatal window, which is the post-wean animal, there's an inability to normalize the NKT cell numbers and normalize sensitivity. But if colonization occurs in the pre-weaned animal, that we're able to normalize this both in a CXCL16 dependent manner through a microbial event that we're not quite clear about yet, but at least one potential microbe that's able to do that is Bacteroides fragilis through the generation of inhibitory sphingolipids in a pathway that happens to be CXCL16 independent. And the same thing in the lung, probably through a different set of microbes, because Ding observed that Bacteroides monocolonization was not able to normalize the NKT cells in the lung, suggesting there's another set of microbes in the lung that are providing this ability to normalize. And this has turned out to be a very interesting theme that's been picked up by others. And since we published that paper, there have been several groups that have taken the notion of utilizing germ-free animals to colonize in those first couple weeks of life and compare the responses to colonization post-weaned to evaluate the biologic plausibility of the relationship to the potential hygiene or microbial hypothesis. For example, Kathy McCoy, working with Andrew McPherson's group, has shown that the same thing happens with IgE. Again, IgE is elevated in germ-free animals and can only be normalized if the animals receive the microbial signals in early life. And I think what's very interesting about this paper in particular is that they needed to have a rather complex consortia of organisms in order to achieve the normalization of the Ig levels. Another good example, or set of examples, are recent papers from Scharschmidt and colleagues, Scharschmick and colleagues, which has shown that the same thing happens for T regulatory cell infiltration, induced Tregs in the skin, and a similar study from the Marsden group, which suggests that the same thing happens in the lung, that again, early life colonization is able to regulate the numbers of Tregs in these two organs, the skin and the lung, but only when conventionalization occurs in early life. And so in terms of my summary, my vision of this is that atopic, as well as numerous complex diseases, in addition, IBD is the poster child here that I've shown you today, I believe and I predict will be shown to originate from an inappropriate microbial exposure during early life through pathways that I believe are developmentally regulated. Our current notion is that NKT cell infiltration into the colon is a developmentally regulated process that microbes are actually having some influence over. And thus, therefore, making it likely that there are some pathways that may be linked to later life sensitivity to environmental events that will require the proper microbial education early in early life, which is a challenge. And therefore, I think it's important for us to identify those pathways that are developmentally regulated, because not all the pathways that are responsive to conventionalization require early life education. And there are many examples. For example, T cells and B cells can easily be normalized when conventionalization occurs in the post-wean state. But I think understanding those pathways that are age-dependent or temporally dependent becomes very, very important as we start to think about the pathophysiologic pathways that they are associated with later in disease. And I think importantly, the environmental triggers that are impinging upon those specific dysregulated environmental pathways. And so therefore, the opportunity is if we can understand those age-regulated events, we may be able to regulate them. But the challenge, obviously, is confirming the existence of these temporally restricted pathways in humans, and thus defining methods to prevent or treat disease, either providing the right exposure in the first instance to generate a normal immune response, as well as understanding those pathways that are triggering those abnormal pathways and inducing disease later in life, and trying to remove those in later life. I have some slides as an example of translating this to humans, but because of time, I'm not going to show it. But I will show the most important slide, which are the people that did the work. I just want to say that most of this work I shared with you was done by Torsten Olzak, working very closely with Ding Ding Yang in Dennis Kasper's lab, who's a very close friend and collaborator. Sung Wano was the chemist who I mentioned. Sebastian Zeissig actually kicked this set of experiments off and has his own lab in Dresden now. And the human studies I was going to tell you were with Etta Freeberger and Willem Lexman. Maybe we could talk about it during the question and answer period. And for that, thank you for this invitation and my opportunity to speak to you.